Now we are going to learn about phage strategies. It's a molecular biology portion. Let's have a chapter outline. Introduction. Then lytic development is divided into two periods. Then lytic development is controlled by a cascade. Two types of regulatory events control the lytic cascade. Phage T7 and T4 genomes show functional clustering. Then lambda immediate early and delayed early genes are needed for both lysogeny and lytic cycle. Then the lytic cycle depends on anti-termination by PN. Then lysogeny is maintained by the lambda repressor protein. The lambda repressor and its operators define the immunity region. The DNA binding form of the lambda repressor is a dimer. The lambda repressor uses a helix turn helix motif to bind DNA. The lambda repressor dimers bind cooperatively to the operator. Then the lambda repressor maintains an autoregulatory circuit. Then cooperative interactions increase the sensitivity of regulation. The C2 and C3 genes are needed to establish lysogeny. A poor promoter requires C2 protein. Then lysogeny requires several events. The CRO repressor is needed for lytic infection. What determines the balance between lysogeny and lytic cycle? So let's have an introduction about this topic. A virus consists of a nucleic acid genome contained in a protein coat. In order to reproduce, the virus must infect a host cell. The typical pattern of an infection is to subvert the functions of the host cell for the purpose of producing a large number of progeny viruses. Viruses that infect bacteria are generally called bacteriophages, often abbreviated as phages or simply lambda, or by the sign represented like this. Usually, a phage infection kills the bacterium. The process by which a phage infects a bacterium, reproduces itself, and then kills its host is called lytic infection. In the typical lytic cycle, the phage DNA or RNA enters the host bacterium. Its genes are transcribed in a set order. The phage genetic material is replicated and the protein components of the phage particles are produced. Finally, the host bacterium is broken open or lysed to release the assembled progeny particles by the process of lysis. For some phages called virulent phages, this is their only strategy for survival. Other phages have a dual existence. They are able to perpetuate themselves via the same sort of lytic cycle in what amounts to an open strategy for producing as many copies of phages as rapidly as possible. They also have an alternate form of existence too, in which the phage genome is present in the bacterial genome in a latent form known as prophage. This form of propagation is called lysogeny, and the infected bacteria is known as lysogens. Phages that follow this pathway are called temperate phages. In a lysogenic bacterium, the prophage is inserted or recombined into the bacterial genome and is inherited in the same way as bacterial genes. The process by which it is converted from an independent phage genome into a prophage, that is, a linear part of the bacterial genome, is described as integration. By virtue of its position of a prophage, a lysogenic bacterium has immunity against infection by other phage particles of the same type. Then immunity is established by a single integrated prophage. So in general, a bacterial genome contains only one copy of a prophage of any particular type. Then transitions occur between the lysogenic and lytic modes of existence. Next figure shows that when a temperate phage produced by a lytic cycle enters a new bacterial host cell, it either repeats the lytic cycle or enters the lysogenic cycle. The outcome depends on the conditions of infection and the genotypes of the phage and the bacterium. So in this picture, you could see lytic development involves the reproduction of phage particles with the destruction of the host bacterium. But lysogenic existence allows the phage genome to be carried as part of bacterial genetic information. So lytic cycle is happening, which involves like progeny phages are released from lysed bacterium, but phaged DNA, bacterial DNA, lysogeny is happening. Like there must be some lysogenization. Like phage DNA is integrated into bacterial genome. Bacteria live happily ever after. The lysogenic bacterium is immune to further infection. In reduction, phage DNA is released and enters lytic cycle. So a prophage is formed from the restrictions of lysogeny by a process called induction. First, the phage DNA is released from the bacterial chromosome by another recombination event called excision. The free DNA then proceeds to the lytic pathway. The alternate forms in which this phages are propagated are determined by the regulation of transcription. Lysogeny is maintained by the interaction of a phage repressor with an operator. The lytic cycle requires a cascade of transcriptional controls. The transition between the two styles is accomplished by the establishment of repression. Lytic cycle to lysogeny or by the relief of repression, induction of lysogeny to lytic phage. These regulatory processes provide a wonderful example of how a series of relatively simple regulatory actions can be built up into complex developmental pathways. Then lytic development is divided into two periods. A phage ineffective cycle is divided into an early period before replication and the late period after the onset of replication. A phage infection generates a pool of progeny phage genomes that replicate and recombine. Phage genomes, by necessity, are small. As with all viruses, they are restricted by the need to package the nucleic acid within the protein code. This limitation dictates many of the viral strategies for reproduction. Typically, a virus takes over the apparatus of the host cell, which then replicates and represses phage genes instead of the bacterial genes. Usually, the phage has genes whose function is to ensure preferential replication of phage DNA. These genes are concerned with the initiation of replication and may even include a new DNA polymerase. So, changes are introduced in the capacity of the host cell to engage in transcription. They involve replacing the RNA polymerase or modifying its capacity for initiation or termination. The result is always the same. Phage mRNA are preferentially transcribed. As far as protein synthesis is concerned, the phage is, for the most part, content to use the host apparatus, redirecting its activities principally by replacing bacterial mRNA with phage mRNA. Lytic development is accomplished by a pathway in which the phage genes are repressed in a particular order. This ensures that the right amount of each component is present at the appropriate time. The cycle can be divided into two general parts illustrated later. Early infection describes the period from entry of the DNA into the start of its replication. Late infection defines the period from the state of replication to the final step of lysing the bacterial cell to release progeny phage particles. In this picture, we could see lytic development takes place by producing phage genomes and protein particles that are assembled into progeny phages. Like phage particles, they are infection. Phage attaches to bacterium. Then DNA injected into the bacterium. Early development is happening. Enzymes for DNA synthesis are made. Replication begins. Late development, genomes, heads and tails are made. DNA packaged into heads, tails attached. Then lysis cell is broken to release progeny phages. The early phase is devoted to the production of enzymes involved in the reproduction of DNA. This includes the enzymes concerned with DNA synthesis, recombination, and sometimes modification. Their activities cause a pool of phage genomes to accumulate. In this pool, genomes are continuously replicating and recombining so that events of a single lytic cycle concern a population of phage genomes. During the late phase, the protein components of the phage particles synthesized. Often many different proteins are needed to make up head and tail structures, so the largest part of the phage genome consists of late functions. In addition to the structural proteins, assembly proteins are needed to help reconstruct the particle, although they are not incorporated into themselves. By the time the structural components are assembling into heads and tails, replication of DNA has reached its maximum rate. The genomes then are inserted into the empty protein heads, tails are added, and the host cell is sliced to allow release of new viral particles. Then lytic component is controlled by a cascade. Key components concepts. The early genes transcribed by host RNA polymerase following infection include or comprise regulators required for expression of the middle set of phage genes. The middle set of genes including regulators to transcribe the later genes. This results in the ordered expression of groups of genes during phage infection. The organization of the phage genetic map often reflects the sequence of lytic development. The concept of the operon is taken to somewhat of an extreme, in which the genes coding for proteins with related functions are clustered to allow their control with the maximum economy. This allows the pathway for lytic development to be controlled with a small number of regulatory switches. The lytic cycle is under positive control so that each group of phage genes can be expressed only when an appropriate signal is given. Next figure shows that the regulatory genes functioning in a cascade in which a gene expressed at one stage is necessary for synthesis of the genes that are expressed at the next stage. Like in this picture, early phage genes are transcribed by host RNA polymerase. Types of gene product: regulator genes, RNA polymerase, sigma factor or anti-termination factor. Then middle, early product causes transcription of middle genes. Then regulator genes, sigma factor or anti-termination factor. Structural genes are their replication enzymes. Then late middle product causes transcription of late genes. Then structural genes are their phage components. So phage lytic development proceeds by a regulatory cascade in which a gene product at each stage is needed for expression of the genes at the next stage. The early part of the first stage of gene expression necessarily relies on the transcription apparatus of host cell. So in general, only a few genes are expressed at this time. Their promoters are indistinguishable from those of host cell genes. The name of this class of genes depend on I'm sorry. The name of this class of genes depends on the phage. In most cases, they are known as early genes. In lambda, phage lambda, they are given the evocative description of immediate early genes. Irrespective of the name, they constitute only a preliminary set of genes representing just the initial part of early period. Sometimes they are exclusively occupied with the transition to the next period. So in all cases, one of these genes always encode a protein, a gene regulator that is necessary for transcription of the next class of genes. This next class of genes in the early stage is known variously as a delayed early or middle gene group. This expression typically starts as soon as regulator protein coded by the early gene is available. Depending on the nature of the control circuit, the initial set of early genes may or may not continue to be expressed at this stage. If control is at transcription initiation, the two events are independent as shown in the next figure, and early genes can be switched off when middle genes are transcribed. If control is at transcription termination, the early genes must continue to be expressed. Often the expression of host genes is reduced. Together the two sets of early genes account for all necessary phage functions except those needed to assemble the particle code itself and to lyse the cell. Early region is there, next region is there, promoter is there, terminator, promoter, early region mRNA, next initiation, late region mRNA. Control at initiation utilizes independent transcription units, each with its own promoter and terminator, which produce independent mRNAs. The transcription units need not be located near one another. The next figure, control at termination requires adjacent units so that transcription can read from the first gene into the next gene. This produces a single mRNA that contains both sets of genes, like promoter, terminator, anti-termination, late region, early region, etc. can be seen. When the replication of phage DNA begins, it is time for the late genes to be expressed. Their transcription at this stage usually is arranged by embedding an additional regulator gene within the previous delayed early or middle set of genes. This regulator may be another anti-terminator factor as in lambda, or it may be another sigma factor such as in bacillus subtilis factor. Lytic infection often falls into the stages just described, beginning with the early genes transcribed by host DNA polymerase. Sometimes the regulators are the only products at this stage. This stage is followed by those genes transcribed under the direction of the regulator produced in the first stage. Most of these genes encode enzymes needed for replication of phage DNA. The final stage consists of genes for phage components, which are transcribed under the direction of a regulator synthesized in the second stage. Then the use of the successive controls in which each set of genes contains a regulator that is necessary for expression of the next set creates a cascade in which groups of genes are turned on and sometimes off at particular times. This means used to construct each phage cascade are different, but the results are similar. Two types of regulatory events control the lytic cascade. Regulator proteins used in phage cascade may sponsor initiation at new phage promoters or cause the host polymerase to read through transcription terminators. At every stage of phage expression, one or more of the active genes is a regulator that is needed for the subsequent phage. The regulator may take the form of new sigma factor that redirects the specificity of the host RNA polymerase of an anti-terminator factor that allows it to read a new group of genes. See the prokaryotic transcription chapter. The following discussion compares the use of switching and initiation or termination to control gene expression. One mechanism for recognizing new phage promoter is to replace the sigma factor of the host enzyme with another factor that redirects the specificity in initiation, as shown with the help of next picture. An alternative is to synthesize a new phage RNA polymerase. In either case, the critical feature that distinguishes the new set of genes is their possession of different promoters from those originally recognized by host RNA polymerase. Next figure shows that the two sets of transcripts are independent. As a consequence, early gene expression can cease after the new sigma factor or polymerase has been produced. Holoenzyme with sigma 70 reconciles one set of promoters. Phage synthesizes new sigma RNA polymerase. Phage sigma factor causes host enzyme to recognize new promoters. Phage RNA polymerase recognizes new set of promoters. A phage may control transcription at initiation either by synthesizing a new sigma factor that replaces the host sigma factor or by synthesizing a new RNA polymerase. Then anti-termination provides an alternative mechanism for phages to control the switch from early genes to the next stage of expression. The use of anti-termination depends on a particular arrangement of genes. Next figure shows that the early genes lie adjacent to the genes that are to be expressed next but are separated from them by terminator sites. If termination is prevented at this sites, the polymerase reads through this into the genes on the other side. So in anti-termination, the same promoters continue to be recognized by RNA polymerase. The new genes are expressed only by extending the RNA chain to form molecules that contain early DNA sequences at the 5' end and the new gene requires at the 3' end. The two types of sequences remain linked. This early gene expression inevitably continues. The regulator gene that controls the switch from immediate early to delayed early expression in phage lambda is identified by mutations in gene N that can transcribe only the immediate early genes. They proceed to further infect a cycle. Later, from the genetic point of view, the mechanisms of new initiation and anti-termination are similar. Both are positive controls in which an early gene product must be made by the phage in order to express the next set of genes. By employing either sigma factor or anti-termination proteins with different specifications, a cascade for gene expression can be constructed. Next topic we will be learning with the help of next video. Thank you for listening.